for lectures 1 and 2, when it comes to the content, y'all will be responsible for reading lecture 1 and 2 material. I wanted to go ahead and make a supplementary PowerPoint and video recording over the PowerPoint just to help y'all out. It's really not a whole lot of information in lecture 1 and 2. Like I said before, we're just picking out the most important things for y'all to memorize. But we'll just do a quick supplementary PowerPoint here. So for lecture 1, this presentation introduces basic molecular biology concepts, foundation of molecular diagnostics, including DNA and RNA structure, chromosomes, and the central dogma, replication, transcription, and translation. And move to the next slide here. We do have a few key terms, including nucleotide, which are building blocks of DNA and RNA, a gene, which is a DNA segment that encodes a functional product. A allele is a variant form of a gene. Exons are the coding portion of a gene. Introns are those in-between parts on the DNA that are non-coding sequences within a gene. Codons are 3 nucleotide sequence in mRNA that specifies a particular amino acid. Replication is DNA duplication prior to cell division, and transcription is DNA to RNA conversion, and translation is RNA-directed protein synthesis. So when it comes to DNA structure, DNA is a double-stranded macromolecule made of nucleotides, which consist of a deoxyribose sugar, a phosphate group, and a nitrogenous base, which is going to be A, T, G, or C, like A being adenine, T being thymine, and so forth. Base pairing includes A to T and G to C in DNA, so A's and T's pair up between the two DNA lengths and stuff, but like between the 5 to 3 and the 3 to 5 ends. You'll see that here in a minute. And then G's and C's hook up with each other, so that's pretty much, they'll only hook up to each other that way in DNA. They stabilize the helical structure, and strands are anti-parallel in a 5 to 3 strand, and then a 3 to 5 strand, so we'll see that here in a minute. So you can see that deoxyribose in the middle there. We've got 2 strands of DNA here on the right. The yellow bits right there is the deoxyribose. I'm sorry, no, that's the nitrogenous base. And then next to it in pink is the deoxyribose. And a phosphate group. Each strand of DNA between these nitrogenous bases are held together by a hydrogen bond. And then RNA structure. RNA is a versatile molecule that decodes and expresses genetic information. And like DNA, RNA contains the sugar ribose and uses uracil in place of thymine. So when it comes to RNA, between the strands, adenine will hook up with uracil. There won't be a thymine. So A and U match together in RNA. RNA is usually single-stranded and exists in multiple forms, including messenger RNA, ribosomal RNA, and transfer RNA. Messenger RNA plays a central role by carrying the genetic code from DNA to ribosomes, where then proteins are synthesized. And then you can see there, it's just single-stranded. On the left, there's RNA, and then on the right, there's DNA. So you'll see the double helix of that. When it comes to chromosome structure, chromosomes are highly organized DNA protein complexes found in the nucleus of eukaryotic cells. Humans have 23 pairs of chromosomes. 22 pairs are autosomes, and then 1 pair is sex chromosomes. Each chromosome has a centromere, telomeres at both ends, and contain numerous genes. Genes may have several alleles and are composed of coding regions and non-coding regions known as exons and introns. An individual's genotype is the set of alleles they carry. And then the phenotype is the observable trait expressed. So there's the chromosome structure there. Nucleic acid function and regulation. So nucleic acids, like DNA, within DNA and RNA, serve as both the storage medium for genetic information and the tools for its expression. The central dogma of molecular biology describes the flow of genetic information from DNA to RNA to protein. Gene expression involves regulated transcription of DNA into RNA, followed by a translation of that RNA into functional proteins. This regulation ensures genes are expressed in the right cells at the right times and quantities. So DNA transcribed to RNA, which is translated to proteins. Replication is known to be semi-conservative, so each new molecule contains 1 parent and 1 new strand. DNA polymerase 3 synthesizes new strands at the replication fork using base pairing rules. So, DNA polymerase 3 adds nucleotides in the 5-3 direction using a parental strand as a template. The leading strand synthesis is continuous, while lagging strand synthesis is discontinuous, forming what's known as Okazaki fragments. And then, there's a visual representation of transcription right there. That RNA polymerase is an enzyme that's going to be creating new DNA. When it comes to translation, it's the process by which ribosomes convert mRNA sequences into functional proteins. Each set of 3 nucleotides in mRNA, called a codon, specifies a particular amino acid. Transfer RNA, also known as tRNA, brings amino acids to the ribosome within a cell, matching its anticodon with the mRNA codon. Translation begins at a start codon, usually AUG, adenine, uracil, and guanine. It ends at a stop codon, which can be UAA, UAG, or UGA. So, total, there's 64 combinations of codons, which code for 21 different amino acids. And then there is a visual representation of translation, with there being that RNA segment there, and you can see that anticodon is picking those nucleotides up or matching up to them in groups of 3 per codon to create an amino acid. When it comes to lecture 2, that one goes over different specimen types in the lab. So really almost anything that can be obtained from a person can be a molecular specimen. There's just, when it comes to certain vendors out there, like certain manufacturers, if you're testing for a panel of like respiratory viruses, they're going to want a sputum or a bronchial washing, or usually sputum. Or say if it's a enterovirus, something that would live within the intestines, they're going to want like a stool sample. So it just depends on what's being tested for, what sample type you're gonna need. So, yeah, wide variety of specimen types are used depending on the clinical question. These include whole blood, serum or plasma, urine, feces, saliva, spinal fluid, joint fluids, and various solid tissues, which we don't really do. That's more of a histology thing. For solid tissues, each type has specific collection requirements and diagnostic value. For example, stool specimens, they want those collected in a certain type of container which has a preservative in them, like Carrie Blair or something like that, rather than it just testing a hard stool. And then when it comes to blood specimens, blood's one of the most commonly used specimens in molecular diagnostics. Venous blood collected by venipuncture is standard. The use of anticoagulants like EDTA or heparin affects the quality of DNA or RNA extracted, which y'all see in y'all's media lab. Hemolysis or red blood cell rupture can introduce free hemoglobin, which inhibits PCR reactions, which can be especially problematic when working with RNA. Special extraction protocols may be needed to reduce this interference. For urine specimens, urine is not typically used for human genomic testing, but it can be valuable for detecting pathogens such as chlamydia and BK virus. First void urine is optimal for in cases of like inflammation of the urethra, while midstream samples better reflect bladder pathology, so like the cells, like the transitional epithelial cells. Proper collection technique, like using a sterile container and cleaning the area, minimizes contamination. For fecal specimens, fecal testing is shifted toward molecular diagnostics for pathogens and early colorectal cancer detection. Hemoglobin present in bloody stools may inhibit amplification steps, requiring additional preparation. DNA from feces may reflect the genetic status of colonic cells, serving as a potential biomarker for cancer screening. And preservation is key with it. A lot of times these days, they want it in like a Carrie Blair preservative. If that's not completely possible, depending on the site, like say it's collected at a nursing home, they want it refrigerated. In fact, for the stool specimens, a lot of times in the hospital they wouldn't have the Carrie Blair with them up on the floor to put the stool in, and so we would just put the sample in a Carrie Blair ourselves, because that's what our instrument was validated to work with, pretty much stool in Carrie Blair, not just straight stool. When it comes to CSF, it's commonly tested to identify infections and malignancies. It typically is collected via lumbar puncture in 3 or more sterile tubes, usually 4. The second tube is usually reserved for micro, including molecular diagnostics. CSF has a low cell count unless there's infection or disease, or if there's a traumatic tap. So careful handling is crucial. For synovial fluids, they are analyzed for infectious causes of arthritis, including Lyme disease and staphylococcal infections. They're obtained via arthrocentesis. Samples should be collected in sterile tubes and evaluated microscopically and molecularly. EDA tubes are preferred for cell counts, while plain tubes, like red top tubes that are known to be sterile, are used for culture and molecular analysis. So if it doesn't have gel in it and it's a red top tube, usually those are supposedly sterile. Amniotic fluid is connected through amniocentesis for prenatal genetic diagnostics. Conditions such as Tay-Sachs or thalassemia can be tested using DNA from this fluid. Since the cell count is often low, culturing is usually required to obtain enough nucleic acid for testing. Gestational timing and fluid appearance also impacts analysis, so really bloody ones can inhibit amplification. Chorionic villus sampling. This enables earlier prenatal genetic testing than the amniocentesis procedure. The samples taken from placental villi, which share the fetus's genetic makeup. The sample is examined for quality and grown in culture before molecular testing is performed. The procedure can be done through the cervix or abdomen, depending on placental location. Different types of serous fluids, so ascites, pleural, pericardial. These body cavity fluids are analyzed for infection or malignancy using molecular methods. Collection is done via paracentesis or thoracentesis or pericardiocentesis, depending on what type of fluid they're getting. Pathogens such as Candida, Cryptococcus species, tuberculosis, and Streptococcus can be identified through molecular panels. Buccal cells, so just like cheek cells, like within your mouth, in the walls of your mouth, that's a non-invasive source of DNA, ideal for situations where blood-derived DNA might be misleading, such as post-transfusion or after bone marrow transplantation. Mouthwash rinse provides a higher cell yield than swabbing. Like, for example, if you rinse with a saline solution, that'll help get the cells off of the walls of your mouth. If the person does use swabs, they must be PCR compatible, such as Dacron or Rayon swabs with plastic shafts, because wooden ones can cause problems sometimes. There was the solid tissues, commonly FFPE and OCT preserved. FFPE tissue yields lower quality DNA due to fixation, but is widely used in pathology. This is not really stuff that y'all will have to worry about. OCT preservation avoids fixation and allows for better nucleic acid recovery. Examples include breast tumors or lymphoid tissues analyzed for somatic mutations or receptor status. From the hair and nails, these are usually used in forensic contexts for DNA identification or toxicological screening. However, standardization is limited, and they're not commonly used for routine clinical molecular diagnostics. And then we'll quickly go over quality assurance and quality control in the molecular lab. This was a PowerPoint that came from that Leela Buckingham book that I was able to get. So, pre-analytical error is the consequence of erroneous or misleading results caused by events that occur prior to sample analysis. It's, the most common form of error is usually pre-analytical before it gets to us, and what we have the least control over. But if it's collected in an improper container, that's something we can catch and have them recollect. Got to be properly labeled, of course, to patient identification or methods of identification. So like name, date of birth, or name and MRN number, medical record number. If a specimen's unacceptable, everything's documented, like if you had to call for a recollect, and we never, ever throw anything away. That way we can show them how it's mislabeled if they do come down for it. And then after, like, say, maybe if they've filled out a form, like if it's a precious sample, like a CSF, they can usually have the option to fill out a mislabeled form, pretty much them saying that they take full responsibility for the labeling of the specimen in the lab, like in front of the lab person. So, it's not completely proper, but for those precious specimens that you can't really get back, like CSF, they can fill out a form for that. If it's urine or a blood sample, though, really they just need to go and recollect for that. So, especially when it comes to new sequencing instruments these days, it usually the sample that the lab's going to get is a very minimal amount of sample. So I did this one thing called sequencing.com where they test your whole genome, and so you give them a buccal sample, actually two, one for each wall inside your on the inside of your cheek. And so they're getting a whole bunch of epithelial cells, and they do have to have like a certain amount to be able to do the testing. So sometimes they do come back with inadequate amount of sample, and they would need another sample. When it comes to blood samples that are hemolyzed, that can cause issues with the DNA and RNA yield, and can cause issues with amplification. Sometimes blood kind of gets in the way of the specimen. Like, you can kind of think of it like that. Especially if you're like looking for pathogens, that's just like an interfering substance almost, the blood. Like it's in the way. It's less likely that you're going to detect what you want to detect if it's there. Solid tissues are best analyzed from fresh or frozen tissues, something we won't have to worry about. They'll collect those types of things in surgery, and usually these things go straight to the pathologist. We don't mess with it. Many times they are put in formalin. Specimens for molecular testing continued. The quality of nucleic acid from fixed tissue depends on the fixing process and the fixative used. So when it comes to blood samples, certain color top tubes are going to be appropriate for certain molecular tests. Heparin is something that is can cause a lot of problems with molecular testing. A lot of times it's honestly going to be red top tubes for these blood samples, or I mean they do other color top tubes, but it just depends on what it is that the doctor's wanting us to test for. And these type of things, like say if it's to send out, we can look up on LabCorp what the test was and what the specimen requirements are. That way we know exactly what type of specimen we need to send to them so it doesn't get rejected. So I'm not going to really go over this too much. But we do want samples for molecular testing to get to the lab as soon as possible. There are these things that you'll read about in the lecture material called RNAs. RNases are real bad about breaking down RNA segments. They're almost ubiquitous in nature. They're all over the place. And so what we were talking about with the whole unidirectional workflow, having that unidirectional workflow helps us not introduce RNases in the environment into that pre-PCR area. So we don't want to go back and forth between the pre-PCR area and the post-PCR area. It's just really, really easy to contaminate your samples and reagents and all that kind of stuff. And many times we work with Event Hood for pre-PCR area. So this is kind of some review from 1201. Of course, there's proficiency testing for any type of reportable test that's done in the lab. So, if a place does blood culture pathogen testing, like multiplex, like with the BioFire or whatever, they are going to have some unknowns sent to them to make sure that that lab is detecting what they're supposed to be detecting. So let's kind of go through this. A lot of this is sort of extra. Alright, so analyte-specific reagents are probes, primers, antibodies, or other test components that detect a specific target. That'll go more in depth in the lecture material. Yeah, most are not subject to FDA. There's a lot of non-standardized stuff going on in molecular, which is becoming more standardized by the FDA. But since it's a relatively newer field, it's not quite as standardized yet. But it's a very important field, molecular diagnostics.
In a quarter mile, keep left to stay on I-35 south.
And then stuff that we had to learn at Luminex is there's in vitro analytical testing, there's research use only reagents, and then there's investigational use only reagents. In vitro is what we're going to be working with. So this is stuff where it's been regulated. It's been reviewed by like the FDA to be, supposedly safe for human testing. Of course, there's different hazardous chemicals. When it comes to documentation of results, so things such as the BioFire that we were going over in class, many times, so let's say the doctor ordered a respiratory panel. So that test is going to test for different things like influenza, SARS-CoV-2, Bordetella, just para-influenza, all sorts of stuff from a sputum sample or a nasal washing. And so RSV is another common one. And so within a test kit made by a manufacturer, it's going to test for all sorts of different things within this respiratory panel. If it's not present, it's going to show up as not detected for those things. But if something is detected, like RSV, next to that, it will show detected. And a lot of times it won't auto-validate on the computer, but it will, like from the instrument, interface over to the LIS system, where you can then manually review it to make sure everything is there, everything matches, and then you can verify it in the computer so that the doctor or the nurses can see it. And if it's positive, certain ones are considered critical. Proper specimen handling is required for accurate test results. So however it's supposed to be collected, it's really important to meet the conditions for that. Like, say if it has to be immediately refrigerated or frozen or just stay at room temperature, it's important to hold to that. And if there's any certain additives that are causes for rejection, make sure it gets recollected. So that's pretty much all of that. So that takes care of week 1, just supplemental information. Like I was saying before, y'all need to make sure to read the lecture 1 and lecture 2, do your media lab, do all your quizzes, and to help with the quizzes for lecture 1 and lecture 2, it's very much in your best interest to fill out those study guides, because everything in that study guide, whatever's on the quiz, will be directly related to those study guides. Y'all just make sure y'all do y'all's quizzes and media lab homework before Tuesday at 9:30. Also, make sure you do the syllabus acknowledgement agreement as well. Y'all just holler at me if y'all have got any questions, and we'll have exam 2 this coming Tuesday, followed by the DNA extraction lab.